Good evening and welcome to our webinar. My name is Konstantinos Balaskas. I have a slight technical problem, so you will be hearing me but not seeing me. I want to thank Heidelberg for inviting me to chair this, which seems to be a very exciting session. I'm pleased to be joined by a panel of colleagues and innovators in their fields who will be presenting some cutting-edge research based on imaging. From my part, I would like to introduce slightly the theme of the first presentation that Tjebo Heeren will present. My role as director of the Moorfields Reading Center has involved high-level oversight of the Mactel Study. And so I will be sharing my screen now with you. So the Mactel Study is a quite unique phenomenon in terms of research projects. It is a study that has been going on for quite a number of times, around 15 years. And although it focuses on a rare disease, it has managed to amass very large numbers of patients across dozens of sites on a global scale. And to give some numbers, you can see at the bottom of this graph the number of sites. Many of them are located in different continents, in Europe, in the US, in Australia, etc. And we've managed to enroll a large number of patients. This is a natural history study, one that will help us understand better the underlying pathophysiology and behavior of this disease and enable a better phenotyping of this disease. So these numbers are impressive for this type of condition. These are the numbers of confirmed patients with Mactel within our cohort. And some sites, of course, are larger and more active in recruitment than others. And these are the total numbers of eyes recruited into the study by stage. And we can see that there is a high proportion of patients in the slightly more advanced stages of the disease that we have recruited. So the Mactel study is taking place in collaboration with the LMRI Institute in San Diego, who is supporting and funding the project. And it has produced very significant output over the years. This is one of the highlights. There was a journal in Retina that was dedicated to projects from the Mactel study. Many of those based on analysis of imaging data performed within the Moorfields Reading Center. And so briefly, I will just mention some of the key outputs of this project. For example, the ability to correlate structure with function by looking at changes in the ellipsoid zone and how they associate with visual acuity, but even more refined associations between structure and function using microperimetry and associations with changes in the outer layers of the retina. It helps also instigate interest in new imaging modalities that had not previously been extensively studied, such as blue light reflectance imaging. And this is just a good example of how imaging, novel approaches to imaging, can help reshape the understanding of a disease entity. And over time, we have progressed very much with the understanding of Mactel, what it actually is, which is a neurodegenerative disease. And in light of this new understanding, now we can interpret in a different fashion the findings that we see in different imaging modalities. And that enabled Tjebo Heeren, who will be our first speaker, to propose a novel classification for Mactel, which is based on modern imaging and our renewed understanding of the underlying pathophysiology of the disease, in what was described by the reviewers of the papers who accepted it as a landmark paper in this field. So I'm very pleased to have Tjebo joining us in the panel today. And I'll hand over to you, Tjebo, to present the findings of your great work.
Thank you very much, Konstantinos, for this very nice introduction. And thank you, Heidelberg, for this great opportunity to talk today. I will talk about Mactel, and I've been invited because I've been doing research about this condition for the last 8 years and actually doing a PhD. I'm a clinical researcher in Moorfields Eye Hospital, originally from Germany, used to work with Frank Holz as well. Now mainly with Kathy Egan in Moorfields. So I'll be talking just a couple of financial disclosures. I'll be talking about Mactel mainly, and as this is an imaging webinar, I think I will focus mainly on the phenotypic findings and not so much about visual symptoms and so on. However, just a quick overview about Mactel. So we believe there's a prevalence of about 1 in 1,000. It's affecting male and female equally. Typical age of onset is actually between 50 and 60. It peaks pretty much at 55 years. And the first symptom is very clearly reading problems. Now, patients typically describe reading as being difficult, and some more like observant patients say the letters are dancing, some letters may be missing. It's typically 1 or 2 or 3 letters in a word, and they also sometimes describe the words as being blurry. What we also found out over the years that they have more problems in low light condition, is a low contrast sensitivity actually mainly, and this is a quote from a patient. He said, "I struggle with small print low light situations, and this is particularly annoying in restaurants, and if he goes to a new restaurant, he looks up the menu online first." So this is just to describe some typical problems, which makes it quite challenging for us, because if a patient comes with 55 and describes reading problem, the first thing that we think about is obviously just presbyopia. But it needs a quite high degree of vigilance here. And I will show how imaging has and will allow us to make a good diagnosis. Now, Konstantinos has already talked a little bit about the Mactel study, and this was actually started in 2005. It's an international collaboration, and it's entirely funded by one family. It's actually the Lowy family, and they have the Lowy Medical Research Institute is also funding my PhD. So I really have to thank them as well. And this world map shows how the sites increased over the years since 2005. Just a small overview. Now I will already go a little bit more into the clinical details, and I will start with some typical phenotypic findings. I will not talk so much about this classic gas and bloody staging from the 90s. It's still quite okay, but we try to get away from this staging, and I will tell you why a little bit later. So there are some really typical clinical features in Mactel that I want to give you as a take home already for later. So one of the most consistent features that we almost always see is that Mactel has a temporal epicenter. Temporal means in a temporal paraphobia. And it's, I hope I mentioned it earlier, it's a bilateral condition. So it's more than 97% is bilateral. Then there's some other features that are maybe a little bit more known. It's like this loss of retinal transparency that we also call graying. This is not a consistently seen feature. It's maybe there in 30 to 40%. This can increase over the time and then decrease again. It may be a sign of a sort of disease activity. It's still not quite clear what graying actually is. And then we have also more characteristic features in Mactel. These are the crystals, which is a very known feature in Mactel. However, it's only present in 30% of cases. So don't rely your diagnosis of Mactel just on the presence of crystals. However, if you've got crystals, you should definitely consider Mactel. Here we also see pigmentation, again, in the temporal parafoveal, more predominant. So this is another, again, I want to point towards this feature here, the temporal epicenter, and the graying here also visible, and the crystals. Another feature are those actually telangiectatic vessels, which is the name-giving feature. And here we see a right angle vessel. It means that the vessel dips into the depth of the retina, and the caliber seems to suddenly get smaller. And this is why they were called right angle vessels. Now, these are more like traditional fundoscopy findings, but we obviously in the last 20 years have done some amazing advancements in imaging. And obviously OCT has provided a lot of new information also about Mactel. So all those OCTs are actually Mactel OCTs. The first one here, where I said very early on the slide, shows a temporal asymmetry. So usually we have the deepest point in the fovea just exactly in the center, but here it's slightly off center. We can be quite certain that this is Mactel because the fellow eye in this case had very clear signs of Mactel. Now this is one of those cases where we would still consider it a so-called unilateral case. So it's like one of those 3% of cases that are so-called unilateral. I say so-called because we see certain changes here in OCT, and it's actually very rare that we cannot see anything in any modality at all on a seemingly unaffected eye. Now, these are other findings that you may have already, that you may be aware of, and I want to particularly point out the intraretinal cystoid spaces. It's not exactly cis, and unfortunately, those cystoid spaces often cause confusion with diseases like diabetic retinopathy, which I want to point that there's no retinal thickening really, and there's this famous ILM drape over the intraretinal cystoid space. And so if you see a thing like that, please consider Mactel. And this is important because you don't want to give anti-VEGF in this case. Anti-VEGF is potentially harmful in those cases and could, this is, I will just, come towards this later again. Some more OCT imaging. And these are later stages. Why do I say later stages in this case? Because the outer retina is also affected. There's a loss of photoreceptors. In this case, the last case, there's an old neovascularization. We've got a subretinal fibrosis and also loss of outer retina. Good. So this is OCT findings, and OCT is actually a very important imaging modality for us, and I would personally be able to make the diagnosis of Mactel based on OCT in probably 95% of cases. However, I've got 8 years experience of Mactel, so please, if you're not that confident, that is also fine. And we've got more imaging modalities actually, and here we've got fundus autofluorescence, which also helps a lot. And again, I want to point towards this temporal epicenter. We've got findings like increased or decreased autofluorescence, which is mainly in the temporal parafovea, whereas we do not really see a lot in the color fundus. And in those cases, I struggle even to see it fundoscopically, even though I've seen personally more than 400 people with Mactel. However, I would still struggle to always see fundoscopic findings. And that's another thing that in fundoscopy, we often oversee earlier stages of Mactel. Now, these are just other examples for findings in fundus autofluorescence. We see a loss of autofluorescence if there's like pigment plug, or here there's this fibrosis. This is an old neovascularization. And we can actually go ahead and analyze this a little bit better. And this is how then research would be done, that we actually compare on the location the exact changes on autofluorescence, then later blue light reflectance. I will present this a little bit later. And macular pigment measurement, how this relates to OCT. So autofluorescence is not a very simple modality, and that's just to show that there's many features in structural changes that can actually lead to different aspects in fundus autofluorescence. One important one is actually the loss or redistribution of macular pigment, which is maybe a pathognomonic feature in Mactel. Was first described around 2008 from Peter Tjebo Heeren, who found that there's a loss or a reduction in central macular pigment, and that's very interesting because it makes very typical pattern of redistribution of this. And how do we measure that? We measure it with dual wavelength autofluorescence, and this is with a green light autofluorescence and blue light autofluorescence, and there's some complicated physical formulas that we with this we can actually then calculate the optical density of macular pigment, because it absorbs blue light rather than green light. But the lipofuscin, which then is excited, then will be it similarly fluoresces with both wavelengths. That's the principle behind it. Now, most of you will not have this amazing tool, which is actually also a Heidelberg tool, by the way, but you may have blue light reflectance, which shows this very, very similarly, and is a very, very good imaging tool to allow this visualization of the redistribution of macular pigment. It's not, it makes an increased blue light reflectance in this oval shape. And whenever there's a doubt about the possible diagnosis, this is actually one imaging modality that we also use to confirm or reject the diagnosis. Now, Konstantinos has already mentioned that we recently actually were able to publish this article about where we defined the Mactel area and the late stage of Mactel. And it's really hot off the press, as you can see here. It's actually a print proof because it's still in print. Peter Tjebo Heeren was the lead, and Konstantinos was also involved. So this is very, very brand new information now. So what we did from the entire cohort of the Mactel study that I showed you earlier, we had 2,000, actually more than 2,000 patients. And we looked at the patients that, firstly at the visual acuity distribution, which you can see here. And we looked at all patients with very low visual acuity below 2200. And the first thing to point out is actually that there are not many people with such low visual acuity. So that's one major finding of the study, that Mactel has not a very bad prognosis for vision. People may be quite disturbed and also and hindered in their daily living, their daily activities. However, it will lead to legal blindness in only a very, very small percentage of cases. That people get bilateral legal blindness is actually less than 3%. And this is a really nice thing to give away to the patients, to tell them. And it's like people are really, really relieved if you can tell them that. So that's just the background. So we looked at the people with really low visual acuity, because we assumed those would be the people with a later stage. And having looked at that, we looked at the clinical features, and then we measured the dimension of the retinal changes that we found. Actually, in this case, we used fluorescein angiography because it was the most consistently available imaging modality. And we saw that if we would have a relative distance from the optic disc margin to the fovea, we could use that as a size, as a scaled size in the fundus. And the Mactel area almost never exceeds one distance between optic disc margin and fovea. And the height is 0.8 of it. So it's an oval shape, which we knew already, but now we've got some scientific evidence for it as well. And those people who had very low visual acuity, we looked at them particularly, and we found that more than half of them actually had simply large photoreceptor atrophy without anything else. Now I want to come back to the staging that Gas proposed in 1993, that it was stage 1 to 5, which suggests that everyone will develop stage 5 in the end, which is a neovascularization. But this is not the case. Actually, most people simply develop photoreceptor atrophy. And this is, therefore, we can say that photoreceptor atrophy actually defines the end stage of the disease. And this is how imaging actually helped us in this case to make this, to make this decision. There are complications. Now, this is a very extreme case of a neovascularization. This is actually only a handful of patients who develop this kind of neovascularization in our whole cohort. We know that this is Mactel because we have got historic images about this. There are other things that we can see maybe a little bit more frequently, and one of the most relevant, I would think, is the subfoveal fluid. It's kind of CSR-like, and indeed, there are other features that are very similar to CSR, like the thickened choroid, for example. If you see something like that without any other sign of neovascularization, you don't need to inject anti-VEGF. So there's macular hold, which is a rare complication, but you can see here an atrophy of the outer retinal layers. It looks different than a simple macular hold. I hope I'm not too much over time. I get some Mactel take-home measures. It's maybe more often than you think. It causes mainly reading problems, especially in low light condition. Is bilateral condition with a temporal epicenter. Photoreceptor atrophy and not neovascularization defines the end stage of the disease, and it's limited to the Mactel area. There are some more messages. I personally find OCT key for diagnosis, and all other modalities help. There's currently no treatment. CNTF is under investigation with ciliary neurotrophic factor, and please inject anti-VEGF only if there are signs of active neovascularization. This is intraretinal fluid, hemorrhage, and subretinal fibrosis combined with subretinal hyperreflectivity on OCT. So thank you very much, and especially to those people here on the slide, and I will pass back.
So I'll just introduce myself. I'm Giovanni Montezano. I'm a research fellow in Professor David Crabb's lab, and for this presentation, I just wanted to show you a quick overview of the work that we've been doing to relate structural and functional measurements in intermediate AMD, especially in macular drusen. This work was mainly supported and financed by the MacT project, although the data don't come actually from the MacT project because that was somewhat like a sluggish recruitment in the whole process. So the main feature that we're going to focus on for this study, and one of the hallmarks of the disease, is actually the presence of macular drusen. And these macular drusen are made of extracellular lipids, extracellular debris, lipids, and proteins that accumulate between the retinal pigment epithelium and the Bruch's membrane. And they form these dome-shaped bulges between the RPE and the Bruch's membrane. And you can see this is the typical aspect that they have at fundus inspection. And this is a cross-section taken with an OCT, and you can see that you can appreciate the dome shape quite well, and it reproduces fairly well what we see in histology. So the main objective of our study was to, again, relate structural measurements and functional measurements. And for structure, we used two different types of imaging modalities, autofluorescence and spectral domain optical coherence tomography. both types of images were taken with the Spectralis SD-OCT and HRA device. And for function, we actually performed a microperimetric test, which I will explain what it is in a few slides, in both mesopic and scotopic conditions. So as I said, the dataset doesn't come from the MacT project, but it comes from a different study, which is called the Northern Ireland Sensory Aging Study 2. And it is basically a population-based data collection that included more than 400 participants. For our specific analysis, we only included people that had a complete set of structural and functional data that we needed to perform our analysis. And we included both healthy subjects and patients with AMD, where by AMD we just meant people that had macular drusen and didn't have an end-stage disease, so no geographic atrophy and no choroidal neovascularization. And as you can see, people that met the actual inclusion criteria were much less than the overall population, but this is expected because this was not an enriched population. It was mostly just extracting people that we needed from a population-based study. So as I said, the autofluorescence was acquired with the Spectralis system, but the main feature of that we exploited of the device was the fact that we could use two different wavelengths to acquire autofluorescence, as Tjebo has just explained in his slides. And the main difference between these two modalities is the fact that green autofluorescence, so the green light, is much less absorbed by the macular pigment. So basically, it almost goes through it unscathed. And the benefit of this is that if you want to see how the light diffusion in the RPE is distributed, and if you have patches of atrophy or of increased autofluorescence, this one will be much less masked by the macular pigment, and so it is less affected by this. What in our case is an artifact. In some cases, might not be an artifact; it might be used for information. But in our case, we were not interested in specifically in the macular pigment, so we used the green autofluorescence to eliminate this problem. Regarding the OCT imaging, we mainly extracted two metrics from the images. The first one was the RPE elevation, which is very simply put, it's just the distance between the red line here and the blue line here, the red line represents the outline, the segmentation of the RPE, and the blue line represents the segmentation of the Brooks membrane. And this is effectively just a surrogate of macular thickness, but doesn't require that we identify individual drusen. So it's just so that's why we're not referring to as macular thickness, because we didn't identify single individual drusen, but we instead just calculated the elevation of the RPE over the Brooks membrane. The other metric that we used, what is called photoreceptor reflectivity, and it is exactly what it sounds like. But before I explain how we actually calculated it, I wanted to show you that this image that you're looking at, which you're used to looking at, is not what the actual OCT data look like. And the original image looks more like this. So it's much darker, and of course this would be pretty much useless for any clinical application because you cannot see most of the layers. But the advantage of this type of visualization is the fact that you are actually seeing what the reflectivity of each individual layer is, and you can see that the photoreceptors and the RPE are much more reflective than the rest. And then the good thing is that, of course, this is a physical measurement of the reflectivity, and this is what we're going to be using for our analysis. So what we wanted to see was actually the reflectivity in this tiny slab just above the RPE, which should include more or less the photoreceptor layer. And the way we calculated this photoreceptor reflectivity index was, for each column, we took the sum of values from the red line downwards and from the blue line downwards, and then we took the ratio of the two signals. And as you can see very intuitively, where you have less reflective photoreceptor layers, you can see that you have these dips in the reflectivity index. So basically it tells you where the photoreceptors are less reflective. If you now perform these analysis on all the B scans that we had available for each for each macular scan, and you stack them next to each other, you can create these nice looking maps of the RPE elevation that shows you where the drusen are, and another map of the photoreceptor reflectivity. And you can use these maps to do your structure function analysis. As I mentioned before, on the functional side, we perform a test called microperimetry, and it is essentially just a perimetric test. So basically, we shine lights of different intensities, and we ask the patient to plug up when they see the stimulus by pressing a button. The main difference with traditional microperimetry is that we usually are focused on a much narrower region of the visual space, which is more focused on the center, so within 10 degrees usually, and the locations that we test are much more closely spaced, so they're very near to each other, So what we need to do, what we need to make sure is that we are able to be as precise as we can when we are measuring the sensitivity at any given retinal location. So on the left, I don't know if you can appreciate the animation, but basically on the left-hand side, you can see what would happen if we didn't account for eye movements. So the projection would stay still, but the eye would move behind it, and basically this would make our measurements very imprecise. So what microperimetry does, it does a live compensation of eye movements by continuous imaging, and keeps the tested locations, the perimetric grid, locked onto the retina, so that it basically chases the eye around. So you can achieve much better accuracy when you're testing locations, and you're sure they always test in the same location. As I said, we use two different types of microperimetry, the mesopic microperimetry and the scotopic microperimetry, and they have two different properties. They are very similar, but the main important difference is actually in the name, is the fact that the background is mesopic in the mesopic test, and is basically pitch black in the scotopic test. The other difference is that for the mesopic test, we use essentially white light. And for the scotopic test, we actually perform two different tests, one with a red stimulus and one with a cyan stimulus. And the reason why we do this is because the red stimulus is supposed to target more the cone component of the scotopic response, and the cyan stimulus is supposed to target more the rod component of the scotopic response. So we are able to monitor the functionality of both types of photoreceptors. So the main problem, as you can see, is that we have these tests will produce very different results. So in order to be able to compare one test with another test in terms of structure function relationship, we need to make things comparable. So what we did, we used the healthy cohort to estimate a normative model, so that we could, instead of using the raw sensitivities for each location, we use the deviation from normal. So we basically quantify the defect of each location compared to a normative reference, adjust for age and eccentricity to see how much loss of sensitivity there was at each location. And this is the normative model for the mesopic test, for the red scotopic test, and for the cyan scotopic test. So this picture is a bit unusual, and the reason why you have this drop off near the fovea and sensitivity is actually explained by two main reasons, by two main features of the visual system. So the first one, and the most important one, is that the density of rods drop off quite rapidly towards the fovea and eventually goes to zero, and this is reflected by the scotopic cyan test, which is mainly targeting rods. And the other thing is that the cyan being towards the blue end of the spectrum is actually absorbed by the macular pigment. And actually, since we acquired both green autofluorescence and blue autofluorescence, we could calculate the macular pigment optical density, and we could use this complicated formula that you need you don't need to pay attention to compensate for the macular pigment absorption. But the majority of this effect is due to the drop-off in the density of rods. So now that we have everything laid down, we can use, we can match the different pictures coming from different devices, and we can actually report our tested locations from the microperimetric test onto the structural maps. And here you can see what you have on the far right. You have the green autofluorescence, and here you have the near infrared image that the SPECTRALIS uses to track the eye during the acquisition of the OCT scans in a similar way to what microperimetry does. So it's kind of the same concept applied both to imaging and functional testing. And of course, once you have tracked your points onto the near infrared image, you can also translate them to the structural maps. This is the photoreceptor reflectivity map, and this is the RPE elevation map. We are here I've called it drusen thickness, but it is essentially the same thing. So now moving on to the rather disappointing results, we can see that the structure-function relationship is actually fairly weak, and this was kind of expected. These are the results from univariable models, so it's just one structural measurements against the sensitivity loss for each individual test, and all the structural metrics have been transformed in the log scale to match the scale of decibels that is the one that we use to measure sensitivity. And as you can see, the strength of the relationship, as shown by the R-squared, is not very strong. Most of them are significant, but the relationship is not very strong. And this is kind of expected because we were not expecting to see nothing more than very, very early damage. And if you only have really tiny deviation from the normal function, then you cannot really measure, and you don't have a lot of variability in your functional measurement to that would match your structural changes. Essentially, we found what we were expecting to find, that higher RPE elevation is usually associated with lower sensitivities. The best functional test was actually the mesopic test. A lower reflectivity of the photoreceptor layer is actually associated with lower sensitivity, which was also expected because we think that the reflectivity of the photoreceptors represent the health of the photoreceptors. And the green autofluorescence pretty much mimicked what the photoreceptor reflectivity was telling us, and we think this is mostly due to the fact that a decrease in autofluorescence means that the photoreceptors and the RPE on top of the drusen are less healthy, and so the autofluorescence decreases. These are the results of a more complex model that basically takes all the structural measurements into account to create one multivariable model. And what we see is that, again, rather disappointing results in the sense that the best test was by far the mesopic test, but we can see that the prediction is quite poor from the R-squared. In these graphs, what you have the predicted loss from the model, from the multivariable model, and the real loss that we've seen in the patients, in the subjects. And the black line at the center represents the line of equivalence. So basically, if we had perfect agreement between the prediction and the real data, you would see that all the points will be lying on this thick black line. The reason why, again, this speaks to the fact that the main reason why we observe the poor structure-function relationship is because the damage is very mild. And you can see, in fact, that most of the noise around the prediction is actually compatible with what the test-retest of microperimetry is from the literature. So you can see that here the dashed lines are the limits of test-retest variability. So it means that if you were to perform the test twice, you would expect a variability that is approximately this large. Another thing that we need to consider is the fact that most of the locations that we tested with microperimetry were actually not affected by drusen, because we didn't target the drusen. We didn't target our locations to be specifically where the drusen are. And so we, as a further test, we isolated those locations that corresponded to regions where the RPE elevation was higher than normal. And what we found is that the structure-function relationship is improved, which was expected, but still it is an important result because it means that using both structure and function can be used to follow up and to predict how things are going to go in the locations that are already affected, which are the ones that are more likely to show any progression. Again, the mesopic test was by far the best test of the three that we used. So in conclusion, this is my final slide. Structure-function relationship in people with drusen is actually weak, and it's mostly due to the initial damage. It is much stronger with the mesopic test. This might be due specifically in our data set to the limited dynamic range that we had in the scotopic test, because we used an old version of the software for the Maya, for the scotopic Maya, that only had 20 decibel in dynamic range, whereas the mesopic test has 32 decibels in dynamic range. But this is unlikely to be the explanation for our findings, because we did observe any ceiling effect, so the data were not clipped upwards even in healthy subjects. And finally, the structure-function relationship is much stronger in areas where we actually have drusen, which is useful to know because it means that we can use and combine information to improve the detection of progression in these patients. So I would like to thank all the people that helped me with this project, including Beth, which is our PhD student, Ruth Hogg, who provided data, and my supervisor, Professor David Crabb, and all my colleagues in the lab. Thank you.
So in the interest of time, I will introduce our next speaker, Professor Francesca Cordeiro, who is a chair and professor of ophthalmology at Imperial College London and a consultant in the Western Eye, and has a keen academic interest in translational research, trying to apply molecular and biological approaches to the treatment or pathogenesis of retinal degenerative diseases, but also glaucoma, Alzheimer's, Parkinson's, and diabetes. And today, Francesca will give us an overview of her very exciting work on the DARC project, which aims to help early detection of glaucoma, which is a major priority for healthcare services, for glaucoma services. So thank you very much, Francesca.
Thank you very much, Connie, and thanks very much to Heidelberg, especially Emily. So I'm going to be covering a little bit about the DARC technology, but more specifically our most recent publication, which came out in Expert Review of Molecular Diagnostics last month. And these are my disclosures, in particular for this, Heidelberg and Novi Limited. So the whole technology is centered around the detection of apoptosing retinal cells. And we've been talking about it for a little while. This was our first publication back in 2004. And it's based on the fact that any cell membrane, when it undergoes apoptosis or stress, changes its formation. So that phosphatidylserine lipids, which normally live on the inside, go to the outside. And because they're exposed on the outer cell membrane, annexin 5, which is our biomarker, actually binds to the exposed phosphatidylserine, and because it's fluorescently tagged, you can see it as white spots. So we've been working on this for a little while. We've been using it to check therapies for different diseases. I'm a glaucoma person primarily, so we've got most of this data on glaucoma, but you also have it in AMD, optic neuropathy, and Alzheimer's, Parkinson's, and retinal toxicity. And it's led to this rather unique collaboration between UCL, Imperial, and more recently Novi, though the study we are presenting today specifically has been generously sponsored by the Wellcome Trust funding. This is a very quick overview from the paper from phase 1, which shows you this is a bit like injecting fluorescein sodium or ICGA at the same time as capturing images on a SPECTRALIS. And in 2017, we published our first paper from the phase 1 trial, which showed that it was possible to pick up a single retinal neuronal cell apoptosis in vivo, there was a hint that there was an increased activity in glaucoma disease. But what was very interesting in the post hoc analysis, we were able to show that the dark count, which is the number of white spots, was able to predict those patients that went on to have an increasing rate of progression. And we said this was very important because it suggested that DARC technology could be a promising new biomarker which could lead to early detection of neurodegeneration and prompt treatment response. And we were coming from here, so you all be aware of this idea that you can have a surrogate endpoint and a biomarker. If you think something like stroke, you have increased blood pressure as your surrogate endpoint. We were wondering if DARC could be the surrogate endpoint for true glaucoma. And that went on to the phase 2 clinical trial, which took place in 2017. This was a trial which built on the phase 1, but the primary objective here was to evaluate the efficacy of DARC using just a single dose of the biomarker. But in addition to glaucoma, we increased the number of diseases. So there was glaucoma, AMD, a model of Alzheimer's and optic neuritis, but also we had a much greater number of subjects. So the phase 1 had only got 16 subjects, of which 8 had glaucoma. The secondary objective of this was still to look at the safety and tolerability. And this, I think, is the first real slide I've been able to show you. This is the actual spots that you see in a human retina with glaucoma. So the phase 2 clinical trial was quite a large one at the Western Eye, the iCorg unit at the Western Eye Hospital. We had 113 patients. Each of these patients underwent a single assessment with DARC. 40 of those were controls, healthy volunteers. We had 16 Downs, 18 optic neuritis, but more specifically for this project, 20 glaucoma patients. And we recorded using the ICGA settings for SLO, the baseline 50 minutes, 2-hour and 4-hour photos. So those photos were initially first presented to a set of manual observers, and it gave them a simple task. They had to look at the same lighting conditions on the same computer and see if they could identify single white spots on a human retina. So these were presented to them. 5 masked operators, they used image tray to identify the spots, and then we had to see how they did. Now this is an example. This is an eye looked at by manual observer A. Same eye looked at by B, by C, by D, and by E. Another way of looking at this is the agreement between them. So this is the total number of spots ever seen by any observer. This is when 2 or more observers agree, and this is when 5 agree. And you can have a look at this graph showing that actually the agreement between observers was not that great. Agreement of alphas was 0.51. However, we did need a ground truth. And so our ground truth was where more than 2 observers agreed, because that gave us more than 50% of agreement. So going back to the phase 2 trial, what we were specifically looking at here was a way of objectively and automatically assessing the DARC count. And so first of all, we wanted to train an artificial aided algorithm using convolutional neural network with just using the database of the healthy volunteers, and then we were able to test it on the glaucoma volunteers. So initially 50% of the controls were used for training, the other 50% was used for testing, and then the ultimate test was done in our glaucoma cohort, so untouched in terms of the actual CNN training, where we were able to look at those that were stable, and 18 months later assess actually whether those patients went on to progress. And that definition of progression was just very simple. It was looking at the OCT, the global RNFL ring, 18 months down the line, where the rate of progression was significant but greater than what one would expect for age. That's greater than 1 micron per year. So this is a little animation made by my friends at Novi showing you how the algorithm works. So first the image is aligned and processed. Then using a template matching method, we were able to see if we could identify dark spots. With the candidate spots, we were able to then train using the 2 agree as shown in the green circles. And then the CNN was able to automatically extract those features that identified the true dark spots. So you ended up with true positives where manual and the CNN agree, false positives where it was not seen by the manual, and then finally where it was seen only by the manual. So this developed algorithm increased sensitivity and specificity was better than what we had with our manual spots. This is showing you a little bit more slowly here the fact that in a stable patient, the green again is the 2 agree manual observers, and the blue is where the CNN aided algorithm is present. And the same thing is seen here. This is a progressive patient, so there are more spots. When you look at the statistics, this is comparing the observers with the CNN, and very importantly, the AUC as well as the sensitivity and specificity is much higher for the CNN. Looking at the manual versus the CNN, you can see that both are showing a difference that is significant with an increased count in the progressing group, but the counts are higher with the CNN. And that is for the reasons that I think were obvious from showing you the manual observers. So if you set a cutoff of dark at 30, what this means is that all patients who had a dark count of greater than 30 went on to progress 18 months down the line using the OCT. In other words, the positive predictive value of dark when you have a cutoff of 30 is 100%. And this is incredibly unusual when you're talking about biological markers. So we're obviously very, very excited with this finding. So in conclusion, what we've shown is that the CNN-aided algorithm was trained and developed with the manual observers, and that it performed better than 2 agree. So these are the area under the curves of the CNN compared to the 2 agree versus the sensitivity of 0.86 and 0.71, and finally the specificity was greater. In both the CNN and the manual observers, the dark count was higher, significantly higher in the progressing group compared to the stable group. But finally, and perhaps the most important thing from this study, as far as I'm concerned, is the positive predictive value of dark with a cutoff with a dark count of 30 was 100%, so that all patients who had a dark count greater than 30 went on to progress at 18 months. And that's extremely important when you're trying to put forward a biomarker, because you have a clear cutoff. You can say that you are 100% positive that if a patient has this count greater than 30, they will go on to progress rapidly. And in a clinical trial scenario where you're trying to enrich your population, what that means is you can go for a patient population where the dark count is greater than 30 in the realization that those patients are the most likely to show a result of treatment as opposed to using our gold standards. So I have gone quite quickly, and I apologize if it was too quick, but in addition to all the people who helped in the phase 2, I particularly would like to thank the Wellcome Trust who made the study, and in fact the lab to the bench to bedside idea of this whole translational project possible. Thank you.
Thank you very much, Francesca. It's a brilliant talk. So in the interest of time, I will probably allocate one question to each of the panelists, and maybe I'll start with the reverse order, start with Francesca. I have a question for you. Do you see there ever being a non-IV way of introducing the biomarker that might then have a useful application in a primary care screening program?
Absolutely. We've actually already been developed a nasal version, which allows this not to be given intravenously, therefore, and therefore becomes completely non-invasive. We've got a proof of concept in terms of an animal model, but obviously we need to go a little bit further before we can allow it to be given to the primary care. But we're hoping this would be within the next 5 years.
Thank you. A quick question for Giovanni. So the correlation obviously with microperimetry wasn't as good as we would have hoped. Are you aware of any other metrics of vision function that show good promise for achieving better structure-function correlation? Although we all know that structure-function correlation is a very challenging topic by its nature.
Yeah, so there might be, but the major problem is that most of the other functional tests actually are things that are not like perimetry. They tend to provide just one output for the whole eye. So for example, just the simple visual acuity, they will give just a one number that you can relate to a global measure of to global structural measurement, like the drusen volume or the global RPE thickness, or the global photoreceptor reflectivity or whatever. The main thing that makes microperimetry valuable is the fact that you have the topographic correspondence of at each location between the structural and functional metrics. So I don't see so anything that would move along those lines might have a chance to improve things. There is some evidence that contrast sensitivity, for example, might improve, might have a good structure-function relationship, but the main problem with that is, of course, the fact that you don't have the point-wise correlation at each location.
Thank you very much, Giovanni. And last, a question for Tjebo. This is a question from a clinician, I presume. So they're asking, how do you tell the difference between crystals and drusen if you don't do an OCT?
Yeah, so actually I answered this in the chat, but I'm not sure if everyone saw this. So it's a good question. Just as a reminder, crystals are not a necessary feature for the diagnosis of Mactel, and again, only present in roughly 30% of cases. I personally think they are phanoscopically very different than drusen. So they are really like tiny little dots that glisten, and you can really see them like it's very pretty. And they are really clearly located in the inner retina. Sometimes it seems as if they're hovering over the surface rather than in the level of the retinal pigment epithelium. And of course, there are crystalline drusen as well, but this is, firstly, I don't think they are very often. And secondly, if you've got real crystalline drusen, they really look different. They are more like a mother of pearl-like appearance and are much bigger than crystals in Mactel. So it's really, I would say, it's not possible to really confuse crystals in Mactel with drusen. Yeah, they're really different.
Okay, brilliant. Thank you very much. So we are now at 7:30. Unfortunately, we won't have time to cover many more questions. We may be able to address some of those over email, and we can let you enjoy the rest of this beautiful evening tonight. Thank you all. Thanks to the panelists for this brilliant talk. Thanks to Heidelberg for organizing it. And have a great evening, everyone.
Thank you, Con.
Thank you.
Bye-bye.
Thank you. Bye-bye.